Hello everyone, I'm Yvonne, a children's librarian here at the Manchester City Library, and welcome to our Spring STEAM-powered stories. These are story times that are aimed at grades 3 to 6 that have a science, technology, engineering, art, or math component. For copyright reasons, our stories are only going to be read in person at the library, so please visit our website to register for our stories if you would like to join us. In-person attendance also guarantees a craft kit, which will have almost everything you need for our crafts, which we do each session. And we will also do an extra fun activity that we don't get to do together online. Those can be really fun things like slime or some cool moving crafts or something that's just a little bit hard to show you guys here, but we are going to do in person. But I did want to continue posting these videos here so we can do as many crafts and activities to share with as many people as possible. That being said, let's get into this week's STEAM-powered story. Hello everyone, and welcome to this week's STEAM-powered story. Today we are going to be looking at DNA. As usual, I brought a selection of the books that we have here at the Manchester City Library that deal with our topic today. Of course, we have some other books, but I thought that these were just some fun ones to kind of glance through before we get into our activities. So if you find today's crafts and activities interesting and want to learn a little bit more, then you can come to the Manchester City Library and check out some of these fun books. So I hope to see you guys here to learn more about DNA. But before you do that, we do have a few activities planned. So are really hands-on activities, including our looking at DNA and our creation of Play-Doh or slime, we are going to do during our in-person event. Although in our craft kits, I am going to add the instructions on how to look at the strawberry DNA at home if you would like. But we will be doing our activities and crafts here for our last STEAM-powered story. So let's get started with that. Hello everyone, and welcome to our first activity for our looking at DNA STEAM-powered stories. So this one we are actually borrowing a printout from yourgenome.org. They've got a lot of really cool things on information on DNA, so I figured we would borrow some and do it together. So this first one is going to be our origami DNA. First of all, DNA is deoxyribonucleic acid, which you guys may know, and it's basically a long chain of molecules that contains our unique genetic code. And all of this code together is called our genome, which is why the site that we have for some of our activities from is called yourgenome.org. So the DNA holds all of the instructions for making all of the proteins that make up the rest of our body. Now, DNA includes 4 basic building blocks or bases. That is adenine, cytosine, guanine, and thymine. And we basically just shorten those to A, C, G, and T. Now, in DNA, C and G always pair together, and so do A and T. So that's adenine and thymine always go together, and cytosine and guanine always go together. And that is how the combination of these two will create the instructions for the genome. And those 4 bases make up the 2 strands of DNA, which form together in sort of like a twisted ladder that you may or may not have seen before. I will show you an example, and also that is what we are going to be creating with our origami. So this particular origami sheet that I have printed out already has the bases and is in color. There's also a separate sheet through the link that I provided for you guys that you can print out in black and white, or also print out a blank sheet and actually add in the correct bases as you go. Okay, so the first step, of course, is to cut out our origami template. Okay, now we are going to fold this in half. And as we fold, we're going to want to make sure to make nice crisp folds. You can do that by using a fingernail and running it down the fold after you've gotten in position. You can also use like the smooth curve of something plastic or like the cap of a pen or something like that as well, if you don't want to use your fingernails or you don't have much of a fingernail. Okay, so the next step is to hold the paper up so that the thick lines of the diagonal, and then you have the thin lines of the horizontal. And you want to hold that in front of you, right? And then you're going to fold down on the horizontal. So you will see they're going to be facing the thin line. You're going to fold it down and then fold on that thick line. Remember to give it a good crease and fold it back up and repeat. And we're going to do this all the way down our entire origami model. All right, now we've got our origami model all folded along the horizontal. Now we are going to flip over our origami DNA so that it's sort of kind of curved this way. And now we are going to fold over the diagonal just like we did the horizontal. So we're facing this way, we're going to fold it towards us. So we'll actually end up looking at the thick line as we fold it over. We're going to lift that up and we're going to repeat on the next one. And we're going to continue to do that all the way down, okay? Okay, so now we've got our DNA folded along both lines. Now we're going to take the side without letters, so the white side without letters, and we are going to fold that up and over like this, so the letters are going to appear on that side. Okay, and now we are going to flip that over and do it again on this side. Okay, now we're going to unfold those just a little bit so they're not very tight. So the strips with the letters are not folded flat to the side, but just are up a little bit. And already you can see how our paper is starting to twist. So you're going to grab the top here and the bottom here, and we're just sort of going to start to twist them against each other, just kind of the way that the paper wants to go. Okay, you can fold it all the way down into a spiral and lift it up. And here we have the DNA. You can see how we have the two sides of the DNA. We have the C connected to the G, and we have the A connected to T. And so we've got the two strands in this nice twist, which is called a double helix. And so here is our origami DNA model, which I think is so very cool on how it can collapse and open. Hello everyone, and welcome to our second activity on looking at DNA. Again, we are going to be borrowing from yourgenome.org. This time we are looking at the Code Cracker worksheet, and we are going to see if we can figure out what types of proteins the DNA sequences that we are given actually create. So we talked a little bit in the beginning about how DNA codes everything in our bodies and the bodies of other living organisms, and they give instructions, kind of like a recipe on how to make proteins. And proteins do a lot of work in our bodies and in the bodies of other living organisms. They do things like grow hair. They do things like digest food. They do things like make energy. So there's a lot going on. And we are actually going to figure out some things that proteins do in other living organisms by going through this sheet. Okay, so if you guys want, you can download this Code Cracker wheel. I'll also put it up so that you guys can get a screen grab of it if you would like. And then we also have our DNA codes that we're going to go through. Now we're just going to go through the first one together, but you can definitely go through the other ones as well, because it is pretty fun. I just don't want to take up all of your time. Okay, so let's see. All right, so on our DNA code, it looks like the first one we have to decode is ATG. So we are going to start here with A, then we're going to move down to T, and then we're going to move down to G, which means M is our very first clue to our protein. Then we got our second set is going to be AAG. So we have A, and then G, which is going to lead to K. Then we have TCA, which means we go to T, and then we have to go to C, and then we have to find A, just going to lead us out into S. We have GCT. Can you guys find GCT? Got to start here with the G and go to C, and then hit T, which leads us to A for our next protein clue. Got A, T, T, which means we're going to start here with the green A, then red T, and then red T, which is I. And then T, T, A, which is red T, then red T, and then A, which brings us to L. Got A, C, C, which is A, then C, and C again, which is a T. We got G, G, T, which is G, G, T, which brings us to G in the proteins. Okay, we have T, T, G, which is T, T and G over here, which goes to L. We have CTT, which is C, and then T, T, another L, and then TTC, which is T, T, C, which goes to F in the proteins. And finally, we have GTC, which is going to go G, T, C, and V in the proteins. Okay, now we're going to go to our protein cards, and we're going to see if I did all that correctly. All right, so we're looking for a protein that starts with MKS. So, see, it's not this one. It's not that one. Nope. Oh, here we go. MKSAILTGLLFB. So it looks like this is the protein that we found, that we cracked the code of. And it is an antifreeze protein, which stops fish from freezing in icy water, which is really cool. So you've got all of these protein cards that you can crack with your protein sheet. So I hope that you guys enjoy doing that. I would say that if you want to, you can definitely either race a friend or family member to see who can do it fastest, or time yourself and see if you get faster as you go. Okay, so I hope that we learned a little bit about how DNA and proteins work together, and also that you learned some of the really cool things that proteins can do to help animals survive. Hello everyone, and welcome to the craft section of our STEAM-powered stories looking at DNA. So as you guys may know at this point, we will have any craft kits that we have left over from our in-person event waiting for you in the entryway to our children's room here at the Manchester City Library. So come by, pick one of those up while supplies last. This week we are going to be making DNA sequencing bracelets. So we talked here a little bit about DNA and how it's a two-strand helix shape that always pairs the same DNA bases with each other. So you always have adenine and thymine together, and then you have cytosine and guanine together. That's A, T, C, and G, right? So in each of our kits, we are going to have a strip of paper, and this will have one of the two strands of DNA or a part of one of these strands that will be in the organism that is pictured here and labeled on top. So this one is actually sweet oranges. And then you're going to have a second sheet that is going to tell you a little bit about the part of the DNA that you are making and what it does for this organism. Now we have a lot of different types, so when you come and grab one, you're going to get kind of a random one. But here we have sweet oranges. Here we have flesh-eating microbes. And we also just have ones like Homo sapien or humans. So what we're going to do is you're going to grab one of these kits. You're going to have your instructions here. Well, you have actual printed instructions, which also have the strawberry DNA extraction information on the back. And then you're going to have your DNA code that goes with your bracelet kit. Then you're going to have your 2 strands of elastic. And what you're going to want to do is you're going to want to tie a knot in these 2 strands, leaving a little bit of space, right? And what you're going to do is you're going to use the beads in the bag to encode the strip of DNA that you have printed on this label. And then you are going to use the knowledge that you've gained today to create the second strand of DNA just by knowing this information here. Remember that we have thymine, we have guanine, we have cytosine, thymine, and adenine. And remember, you know what these pair up with, right? So thymine, which is here represented with the letter T in a red circle, is always going to pair with adenine, which has the letter A in a green circle, right? So in this list, we are going to start with our 1 strand of DNA, or half of the strand, which we know is going to have the thymine, which is the red bead that matches this. And we know on the other side, on the other half of the DNA strand, it's going to want to pair up with the green bead, which is the adenine. And we're going to build this as we go. So we know that on the one printed here, the next one is the yellow guanine, which I know will want to pair with the blue cytosine. Okay, and we're going to continue building our DNAs with this size that we have, so we'll go make a completed bracelet. All right, let's do it. All right, everyone, thanks so much for joining me for this week's Steam Powered Stories looking at DNA. I want to remind everyone that we are going to be starting a summer reading very soon. You can sign up starting on June 1st, and you can start recording your reading minutes on June 20th. I want to remind everyone that this is going to be our last Steam Powered Story. We are going to be starting a new session called Elementary Engineering with our summer reading program. It is going to be very similar. We are going to still be doing activities and crafts together, but we will be doing it with all elementary grades. So grades 1 through 6 are going to be welcome to elementary engineering. So I hope I will see you guys there. We are going to have it in person. We will have some videos up, although they probably won't be quite as long with all of our activities like we do with Steam Powered Stories, just because there's going to be so much going on this summer. So I hope to see everyone here at the library for summer reading. Until then, bye!